everyone. Try to stay awake in your postprandial joy. So I was asked to talk about biomarkers, which we've been talking about all day. These are some data that I think we had all hoped to see in an organized form a little more quickly, and we're working on that. These will also be presented in a maybe more formalized way at ACR. So this talk is a bit of a data dump, and I've tried to put all the data out as much as I can, so if you want to lean in close, because some of the graphs are a little bit complicated, so that I can show as much as possible. All right, so first I want to acknowledge this was a collaborative, multi-group effort, both through the NIAID, back when I was there before I came to Pittsburgh, a lot of effort and samples from Cincinnati, and a lot of analysis and discussions with the group at Stanford as well. So I thought I'd talk quickly about what the ideal biomarker would do. It would make our jobs very easy. The ideal biomarker would be there before damage has happened, sensitive and specific for the disease that we care about and not other things. It'd correlate with disease severity. It'd be easy to get, like blood or saliva or tears, not something you have to biopsy. It'd be stable, so I could ship it across the world, reproducible and cheap, bridge across species, associated with a mechanism, and tell us how to treat the patient. Basically, the perfect biomarker would do all of these things. And we ask a lot from our biomarkers, and none of them do all of these things, which is why we measure so many things. So summarizing a lot of data, some of which I'll come back to, this is kind of a smattering of the biomarkers we're already using in systemic JIA, MAS, and this related disease HLH, which I'm not going to talk about, but which is kind of lingering in the background. So I won't read to a literate audience, but I'll talk a bit about each of these in turn. So there's S100 calcium binding proteins, IL18, which is kind of the focus of my research lab, soluble CD163, ferritin, neopterin, which we don't measure as much, but is certainly available for us to measure, CXCL9, a marker of interferon gamma activity, and this soluble CD25. Sorry? Neopterin? Yeah. Yeah, I mean, okay. So the experiment that we did used this technology called SomaScan, which I'll introduce briefly. Long and the short of it is that it measures 1,300 plus distinct proteins in one sample of serum and measures them quantitatively. It doesn't use antibodies the way what we normally do with ELISA or Luminex. It uses these DNA molecules called aptamers. And so the experiment we did was to measure serum from these different groups: healthy controls, inactive systemic JMAS, active systemic JMAS. I can't believe I left out the disease of interest. Interstitial lung disease, systemic JA, PAP patients, and then some controls with NOMAD and SAVI and some PAP. So this is the demographics of the groups that I'll show you. There's a difference between the number of samples and the number of patients, because in many patients we have longitudinal samples, and just ratios and ages. The biggest thing to point out is that the lung disease patients are younger, as you might expect. We have a comparator group that has autoimmune or genetic PAP related to GM-CSF, and they're old. These are samples provided by Bruce Trapnew's group. And so this is the cohort. Okay, so let me just orient you to this graph, because I'll be showing a lot of them. So these are the different groups in Violin plots. Each dot represents a different sample, but not necessarily a different patient. The patients in sort of black squares, if you can see them, are actually NLRC4 patients. Oh, okay, very good. So these black dots here are NLRC4 patients, which are integrated into the MAS groups, even though they don't have systemic JIA. These are active NOMAD and active SAVI patients. All of the SAVI patients have lung disease. And this is prior to IL-1 inhibitors or JAK inhibitors, respectively. Thank you to the Goldbach-Mansky lab. And these are the largely adults with autoimmune pulmonary alveolar proteinosis. And these categories of active, inactive, were in the opinion of the referring investigator. We didn't have enough clinical data to say exactly whether they met certain criteria or otherwise. All right, great. So CRP on the left, ferritin on the right. Simply showing this to show you that the categorizations that we used were somewhat valid based on the biomarkers that we know and love. So as you would expect, CRP, this doesn't show up either. In active MAS, everybody has a very high CRP. It's sort of diverse in systemic JI lung disease. It's actually surprisingly, we thought it would be more down here. In inactive systemic JIA, active NOMID, no surprises there. And then ferritin really differentiating best with the active MAS group, which is a little bit circular, but just validating that these aptamers are in large part measuring what we think they're measuring. So real quickly, what are S100 proteins? They're calcium binding proteins. They do different things inside the cells and extracellularly. I'm not going to belabor this. What do we use them for currently? The Munster group has shown nicely that they are somewhat specific for inflammatory diseases like SOJA and FMF, and are helpful in fever of unknown origin. And they are also helpful in inactive or minimally active systemic JIA. S100 proteins may indicate patients who, if they're still elevated, even though the patients look well, associated with a higher, if your MRP, which is the same as S100 proteins, if your MRP is still high, even though you're clinically inactive, you're more likely to flare. So the context of these is that they're somewhat specific for SOJA and FMF, and when they're still elevated, even if the patient looks well, there may be ongoing systemic GA type disease. And it correlates with a bunch of other things. We're not going to go into that. So, oh geez, it didn't. Where are they? I guess we'll, oh, they're there. Nope. This got all out of whack. Okay, well, what I'll tell you is that they weren't particularly helpful in this data set. I'm sorry, this didn't come through properly. So we're moving on to CXCL9 insoluble CD25. This cohort was a bit surprising in that the CXCL9 levels of the active MAS group were not particularly high as measured by aptamer. So we went actually and did a Luminex validation and saw basically the same thing, that the patients that we'd categorized as active MAS, largely based on ferritin, didn't have tremendously high CXCL9 levels. And so even though these patients were categorized as active MAS, they may not have been in the absolute most severe part or acutest part of.
Many of those samples were collected after the first pulse of steroids.
Right, exactly. So
this we know. So 2 pulses of steroid, it takes 2 days or 3 days for CXL9 to go down after pulses.
yeah.
But if you pulse at that, then it would be.
So just to show that our active MAS group is maybe not the most active. And likewise, soluble CD25 was not particularly helpful in this data set, perhaps for the same reason, perhaps because the aptamer is not as sensitive as the Luminex, but also because soluble CD25 is not always so elevated in MAS patients. And so this is just a little background on why we measure CXCL9 as opposed to just measuring interferon gamma directly, and it's largely because the dynamic ranges are so different. So if you look really carefully, you can see differences in interferon gamma level, but the limit of detection is really about here, which means most of your samples are going to be below a limit of detection. Whereas in CXCL9, you can get multiple logs of difference, so it's a much better biomarker in terms of just its dynamics in the blood. And it's a disease activity biomarker. This is from a emapalumab case report showing that a patient with, this is EBV-HLH, you treat with emapalumab and their CXCL9 comes down nicely. And then some of our data, we'll get to that. So then, obviously, near and dear to my heart, and we've talked a lot about IL18. This actually is not measured by aptamer because IL18 was not part of the somologic array. So this is the fluorescence units of a Luminex done in my lab. We get values very similar to what the Cincinnati group has shown. And not a lot of surprises here. Our active MAS group are all very high. The systemic JA lung disease group in pink, now these are all patients in that group they're not categorized by disease activity. Some are very active, some are not. And what I'll try to impress upon you is that their level of IL-18 is out of proportion to their level of disease activity. Quite a wide smattering in the active systemic GIA group, and really even in inactive systemic GIA, also kind of a wide smattering, suggesting, and this is consistent with what other groups have shown, that you don't have to be in the throes of MAS to have high IL-18. We think it has more to do with it being a susceptibility factor to MAS. And as we've shown before, this is the normal range. The autoimmune PAP patients don't have high IL-18, and Nomen and Savi, it's not particularly elevated. This is the endogenous antagonist of IL-18, IL-18 binding protein. These stars here are patients who are on recombinant IL-18 binding protein, which is why their levels are so high. The stars didn't show up. Oh, no. These are just endogenous levels of IL-18 binding protein in some patients with MAS because it goes up quite acutely as an interferon gamma response gene. But the reason I put these in contrast is because you have really quite high levels of IL-18 in the systemic GA group, not particularly high levels of IL-18 binding protein. So ostensibly, there's a lot of bioactive IL-18 floating around.
So in fact, total IL-18 pretty much associates with free IL-18.
Yeah. I have 5 minutes left? All right. We'll go faster. We talked about IL-18. IL-18 is somewhat of a disease activity biomarker. This is actually from Masaki Shimizu's group, showing that during acute flares, it is higher, and in fact, it might come up a little bit before their flare goes, but it does come down. And this blue line that I drew in is sort of not the normal range, but the range of sort of absurdly high IL-18, meaning that this particular systemic GIA patient came down to about 1,000 and was there for quite a long time. And we see this sometimes in our systemic GIA patients. They're not always elevated. And the white background on this disappeared, but this is a bunch of the systemic GIA patients whose IL-18 never normalizes. Oh, there it is. Okay. So this is over the course of multiple years and multiple time points, and their IL-18s are always, this is the normal range. So we did measure free IL-18 in a few MAS patients. This is, again, not done by the aptamer array. Seems to be much more specific for active MAS, where the IL-18 levels, the total IL-18 levels are very high. All right, so coming back to our data set, that's interesting. So this is a principal component analysis of all the samples that were in the aptamer array. And I think one of the first things to notice is that the principal components don't actually explain a ton of the variation. The biggest principal component only explains about 19% of the variation. So there's a lot of variation within the samples that's not being captured here. But along this PC1, you kind of get a gradient that is ostensibly an MAS gradient, where your inactive systemic GIA MAS and your healthy controls are kind of here. Your lung disease patients are kind of further along that, and then your active MAS patients are much further along. And so these are the main contributors to that principal component 1. I'm sorry you can't see that very well, but some of the main things up there are heat shock proteins, signaling protein called 1433, and we'll go through some of these.
Scott, did you do the same exercise only with SGIA, SGIA with mass, SGIA with interstitial lung disease?
Yeah, I mean, it looks basically the same.
Without all the other diseases?
In terms of the number of samples that are in here, very few of these are Savi, NOMID, PAP. Actually, this doesn't even include the autoimmune PAP patients. Yeah, so this is PC2, which is only 8%. Yeah. Right, not a lot that's worth, there's not like a thematic takeaway from PC2. So I don't have those. What's that? Yeah, so I'm not sure PCA is the absolute best way to look at these data. So this is just a volcano plot of the patients with active MAS versus active systemic GIA to see if we can validate old biomarkers and look for new biomarkers of active MAS. And so some of these are pointed out, some of so Ferritin, far and away, leads that list within this data set. But what's interesting are these markers in here, and I'll kind of break out some of these for you. So these are heat shock proteins, HSP 70 and 90, and quite elevated in the active MAS group, and elevated a bit more than we might expect in the MAS with lung disease group, suggesting, as many of you experience clinically, that there's sort of smoldering MAS-like activity in the systemic JMAS patients, and then really not elevated in any of our other scenarios. Heat shock proteins do a lot of things. They are largely chaperones, which means they help with the folding and stability of other proteins. And when they're outside of cells, what they do kind of depends on what they're chaperoning. So if you have a heat shock protein that is in cahoots with another protein and it gets engulfed by a phagocyte, it can assist with antigen presentation. If you have heat shock proteins that are in complex with DNA and they get engulfed, that can cause inflammatory interferon signaling, but it can also even be anti-inflammatory. So these are considered DAMPs, damage-associated macular patterns, and they're targetable. But the effects, at least in the literature, are a little bit pleiotropic. Another interesting thing was a high amount of glycolytic enzymes. So these are, I mean, don't make me go back to glycolysis, but these are essential steps in glycolysis. And again, strongly correlated with the active MAS group, but also a bit elevated in the systemic GA with lung disease group, even out of proportion to active MAS, and not so much the other groups. The interpretation here is these are supposed to be cytosolic enzymes that are fairly high abundance, particularly in cells that are undergoing glycolysis, potentially neutrophils. So the overextended interpretation may be that these are immune cells, maybe neutrophils, that are bursting apart and releasing glycolytic enzymes.
Or reflection of increased metabolic activity.
Well, right.
Because now it's a target in lupus in fact, right?
And in fact, critically important in what Rafaela showed early in the morning about differential regulation of NF-kappa B and MAP kinases.
Right, so activated immune cells upregulate glycolytic enzymes, but they're supposed to stay inside of cells. So that transcriptional upregulation, has to be coupled with some way for them getting out of the cells as well. Because we're not measuring cells here, this is just serum. So there could be processing cell death, but in concert with the HSPs, I think that so the other part, I didn't include signaling molecules, but in the interest of time, I guess we can go back to them. There's a number of intracellular signaling molecules, 1433. Some of these are STAT1 and STAT3. So there's other intracellular things that probably upregulated and activated immune cells, but are getting out into the cytosol. Maybe not, likely not secreted. Okay, so now this is what we wanted to get to, which is the systemic JAMAS, the PAP group versus all the other systemic JA non-PAP groups. And so, this is fold change against p-value. ICAM5 actually comes up highest, but this is MCP1, which we talked a little bit about. This is CCL2. This is a chemokine that recruits monocytes. Okay. Interferon's also a good inducer of MCP1, both type 1 and 2. So I've broken these out into potential lung biomarkers. So this is soluble ICAM5, which is our biggest hit in this group. This is specifically expressed in lung, except for the CNS. If you look at tissue databases, the highest expression of ICAM5 is actually in the CNS, but the next highest is lung. And there is a bit of a spread in the PAP group as well. If anything, it's downregulated in active MAS, so this is very much not an MAS biomarker. Likewise, MMP7, this is a secreted matrix metalloproteinase, quite high in this lung disease group. A bit of a spread in the autoimmune PAP group. Also, some of the SAVI patients a bit elevated. I don't think I touched on this because we're trying to push forward. MMP7 is made by macrophages, and it specifically is one of the proteinases that cleaves ICAM5. So the thought is if you have activated macrophages in the lung, potentially they're cleaving ICAM5 from the lung, and so ICAM5 could be among the biomarkers that we could look at.
Do you see a strong correlation?
Yeah, So they're quite. I mean, you can get. I don't have the R value in front of me, but the samples where they're up are some of the same samples. And then adiponectin is a cytokine chemokine made largely by adipocytes. And again, we kind of don't see it so much in autoimmune PAP. We do see it in SAVI, and again, the systemic GI lung disease patients. There wasn't a strong signature of other adipokines. This could potentially also relate to overall steroid exposure. So whenever you're looking at any of these are just biomarkers, and you can't imply causality. So some of this could be, it's high in these patients because they're exposed to certain drugs more. And we already mentioned CAM5. Okay, so MCP1, we talked about CCL2. It's certainly a little bit more of a spread and up in the active MAS patients, but not nearly as much as in the systemic GI lung disease patients, but also, interestingly, up in the few SAVI patients that we included. And this is a cytochrome P450 reductase. They're in the same slide just because I wanted to show this one. As representative of a number of things that were just as high in the systemic GI lung disease group as in the active MAS group. Again, potentially a marker of tissue damage or upregulation of stress markers within immune cells that then get released upon tissue damage. But getting to sort of a more interesting thing, and then I'll stop, are a number of allergic or eosinophil recruiting chemokines. The most interesting here being eotaxin-1, which is CCL11, and TARC, which stands for thymus and something or other, CCL17. So these two in particular have been associated with allergic chemokine responses. CCL25 is also associated with allergic, but it also can be seen in non-allergic. And again, there's this anti-correlation between especially eotaxin and TARC being particularly high in the lung disease patients, but not so much in the active MAS patients. And so the analysis that we're doing right now is a little bit more careful, but just to show there's some sense when you look longitudinally in specific patients, and this patient might be familiar to you, Rashmi, might be, that these eosinophilic or lung-related chemokines like eotaxin, and I put in ICAM5 here because that was our biggest hit, are somewhat anti-regulated from the MAS cytokines. So here's HSP90 in pink, IL18 up in black, and as you can see longitudinally, it's always crazy high. This y-axis is not just this expression, this is normalized to the median of the healthy controls. So this is how much higher it is than healthy controls at a particular point.
I have a patient right now who I think is on the way to develop this lung disease, and I noticed that her eosinophil count started going up.
So,
but it's not isolated eosinophilia. I see it as a sort of monomylocytoid lineage. Expansion at the expense of a lymphoid lineage. So that's a trend I'm seeing. I don't think it's isolated eosinophilic expansion.
So the point of the ARD, or one of the take-home points of the more recently published Annals of Rheumatic Disease paper, was that many of those patients had lymphopenia and eosinophilia early in their disease course. And so exactly what you're describing is one of the takeaways of that. What they don't have there really are longitudinal data. They've collected sort of snapshots. So one of the things that we're doing right now, and what I spent 4 hours in Dr. Goldberg-Fancy's lab yesterday doing, is combing through and getting the CBCs from these time points to say what was the eosinophil count here, what was the lymphocyte count here. I know, I'm going over. I think, where is Grant? Yeah, yeah, we're working on it.
But again, sort of that it's myeloid expansion. The eosinophilic expansion is a part of the myeloid expansion, and that happens at the expense of the lymphoid lineage.
Yeah, I don't know that.
But the absolute count of lymphocytes decays.
Yeah.
Why are you saying it happens at the expense?
Well, but I see.
There is
Co-regulated.
There is lymphopenia.
Right. Not always. So we're pulling those data out to get the absolute counts.
I see gradual myeloid expansion and gradual contraction of the lymphoid in that patient.
So there's a couple other patients. This is a patient from Boston who the anti-correlation is not quite as good, but you can kind of see HSP comes down, eotaxin comes up.
So another question is,
So these are all untreated nomad. And we have the eosinophil counts from them. I just looked at them yesterday. They weren't particularly high.
It seems to be a sample issue, and I think it goes back to, I think there are other things that contribute that I agree with these small sample groups and the spread that you see. You see that some of those with ILD have actually normal values. They actually overlap very much with the normal control. So the question are the ones that actually drive the mean to be elevated, those that actually have a predisposition to develop
So-
asthma, and is this what we actually pick up in the patients?
The one thing I just wanted to point out, I've made the point that at least eotaxin, but also some of these other ones, anti-correlated with MAS. The few SAVI patients that we have also seem to be high. SAVI was not part of the filtering for how we came up with the volcano plot there. But again, is this cause effect or is this effect cause? And I think that that's a really important thing for us to get to. Are these just related? I know I have a whole minute. That was a long 5 minutes. Okay, this is unfortunately a very sadly deceased patient who probably shows the strongest anti-correlation of these things, HSP and ferritin sort of going up at the same time that these things are coming down and then rising. So suggesting that there are components to this MAS PAP lung disease that are not all MAS. And this is the last patient again, showing some level of anti-correlation. So summarizing, unlike most of our rheumatic diseases where we have a couple of lousy biomarkers, in systemic J and MAS we actually have quite a lot. And it creates a different problem. We almost have a comedy of riches of deciding when should we be using CXCL9 versus neopterin versus IL18 versus S100 proteins versus the 1,300 other things we just measured and need to validate. So we did a high-dimensional near proteomic analysis. The typical biomarkers behaved more or less as expected. CXCL9 was not as robust, but we did Luminex validation. The R-squared values were quite good, meaning that our active MAS patients were probably at least partially treated. We already have a host of biomarkers to differentiate MAS from systemic JIA. And so the addition of heat shock proteins, maybe some of these cytosolic glycolytic enzymes or signaling molecules are, I think, potentially mechanistically interesting. It would be nice to have other patients who are as sick but don't have these kinds of diseases like sepsis to know whether this is just tissue damage in inflammatory disease, period. And we obviously don't know whether any of these things are better than a ferritin to ESR ratio or many of the other things that have been used. And then in PAP versus the others, the IL18 persists. The level of IL18 is still very much out of proportion even when you control for the level of smoldering MAS activity in these patients. We see some lung and macrophage markers like MMP7 and ICAM5, and maybe some eosinophilic chemokines. I put eosinophilic because allergic entails certain things that may not be at work here. You don't necessarily have to have IL4 and 13 to get up regulation of eotaxin. So what are we going to do going forward? Well, one, write the paper. Validate in other samples that these things are real and by other measurements. We're not all going to be doing aptamer arrays to potentially measure these things. My lab tries to do modeling, and so models do what they're going to do. So we're going to try and look in models of MAS to see what happens when you block heat shock proteins and some of the other things that we see there. And then, some of the hypotheses that this data set is meant to generate, are these true in more tissue-directed things? If we think ICAM5 is coming from the lung, it should be even higher in the BAL or in tissue. So with that, I want to thank the Systemic GI Foundation, who many years ago funded this project, and the efforts of people at lots of different institutions, many of whom are in this room. Thank you.